the paper I'm presenting is the expression in the olfactory receptors in different life stages and life histories of wild Atlantic salmon, or Salmo salar, by John Stone et al. 2010 from the Journal of Molecular Ecology. So, brief introduction. We all know homing and imprinting is integral for navigation of anadromous species, the focus of this paper being the Atlantic salmon. And this paper focuses on the expression of those molecular pathways associated with olfaction when Atlantic salmon are identifying odorants found in their natal streams. So if these olfactory receptor genes are so important, the authors hypothesize that they will see a change in olfactory gene expression, and this would be observed between anadromous juveniles and adults and not in the resident population. So for their study, they headed to Newfoundland, Canada, and they compared 3 populations, 2 anadromous and 1 resident. The first anadromous population is from the Exploits River, which is marked in red. The second anadromous population is from the Campbellton River and marked in blue. And the third resident population is marked in by the gray arrow, and it's from Seal Cove Brook on the Avalon Peninsula. This population is super unique in that they are a dwarf population of Atlantic salmon, referred to as the Wananichi, and have been landlocked for over 90 years, I want to say. So their approach to this study was to use the transcriptional expression for a suite of 30 olfactory and odorant receptor genes across life stages of all 3 populations from Canada. The authors aim to quantify these changes in gene expression at different life stages. But how? Lots of methods. First being sample collection, obviously. The second being RNA isolation and cDNA synthesis. The third being qPCR, or quantitative real-time PCR. And of course, a statistical analysis of that qPCR data collection. So the sample collection, they were able to grab a lot of fish. I was really impressed by this. Fishes are hard to get, and they're hard to process. It's a very time-consuming process. So 35 from Exploits River, 27 from the Campbellton River, and 36 from Seal Cove Brook. The 2 anadromous populations were divided among par, smolt, and adults, while the Seal Cove Brook population was only divided into juveniles and adults, and this is simply because they do not migrate, so they do not smolt, and they were only able to be distinguished based on fork length. I just have the utmost appreciation for the way that they were able to collect and process their specimens in such a timely manner, because it is so important when you're dealing with olfactory epithelial tissue to do it quickly and cleanly, because the tissue degrades quickly and it is easily contaminated. So that was really impressive from my perspective as not performing the same research, but something akin to it. So RNA isolation and cDNA synthesis. Dissected heads were placed in RNA later, and rosettes were dissected in the days following the collection. They did their RNA extractions and then degraded any of the residual genomic DNA with a Trizol reagent, and made sure that only the highest quality RNA was used for cDNA synthesis and was based on a really high RNA concentration. So because they used such a strict protocol for what they would include in their study, they ended up taking out several individuals from each of the populations, just based on RNA concentration alone. So what they did end up using was the RNA from 24 fish from the Exploits River, which were 7 par, 8 smolts, and 9 adults. From the Campbellton River, they ended up using 27 fish, 10 par, 10 smolts, and 7 adults. And from the Seal Cove Brook, they used 17 fish, 8 juveniles, and 9 adults. Then they did a primer design and specificity. So the number of gene-specific primers were manually designed using coding sequences and a LegoCalc to calculate annealing temperatures and make sure that there are no secondary structure formations in their complementary DNA. The OLFC, or odorant receptor genes, that were included in the qPCR and tested to ensure that they're the only genes that were expressed in the rosette and biologically active were included in the study. And then they used an EFA1A sub A gene as their control gene for comparison. They obtained this from the EST database. The only caveat I see with this is they don't go into great detail about why they selected this particular gene. When they used the primers, the genes they used were glutamate receptor, odorant receptors, a sockeye olfactory imprinting receptor, and a couple of others, but none that I felt like mirrored this one. I tried to find it in a database, and maybe I just didn't dig deep enough, but I'm curious as to why this gene, because they do talk about this gene among 6 other potentials that they could have used as a control. So that's a curiosity to me. But moving on, they used qPCR, and this method was chosen because they could analyze multiple genes and across multiple individuals in the same qPCR. So really efficient in the way that they analyze their data. And they were limited by RNA that came from the resident population. So when they ran their suite of genes, so their olfactory, the glutamate, all the things that I just mentioned, they tested the integrity of their complementary DNA in duplicate, even though they had such limited amounts of RNA from the resident population. So that gives me a lot of faith in the integrity of their experiment, and in triplicate for those in 2010. And then they ran a statistical analysis based on the data they collected from the qPCR. So again, the anadromous populations were divided into 3 sets, pars, smolts, and adults, while the residents were just divided into juveniles and adults. A one-way ANOVA was used to analyze the variance in a log2 transformation of relative expression, or the RQ ratio, and pairwise comparisons using Tukey's tests. And differences between the means were said to be statistically significant for p-values of less than 0.01. So going over the results, not adults, there's a lot going on here. So basically there's 7 odorant receptor genes that are on the screen. They all have numbers, and those are indicated by A, B, C, after all of those. And what they're finding is that the expression of 2 regions of the control gene remain constant across all individuals and all populations. That's numero uno. Number 2 is the fact that those other genes I mentioned that they used as primers, the sockeye olfactory imprinting gene and another olfactory receptor gene, were excluded from this, from the results in here, because they were creating secondary structures in that complementary DNA. So those were taken out. But of these odorant receptor genes, they found in the exploit salmon river population 5 of them that were upregulated in juveniles compared to adults. And those are in 2.2, 3.1, 13.1, 15.1, and 16.1. And those are indicated in red. So in the paper, you'll be able to decipher the shapes for each of the pars, smolts, and adults. And there's a nice key for that and a better looking key on the second figure. In the Campbellton River population, there were a greater number of differentially express olfactory genes, 17 of them to be exact, with 16 of them being upregulated in comparison to pars in adults, and 10 of them being upregulated in comparison to smolts in adults. So quite the spread, and those are all indicated in blue here. And though they weren't statistically significant for the juvenile and adults in the resident population, you still see upregulation of these genes. And you even see upregulation of these genes in the adults. So not statistically significant, but there is still an increase in these odorant receptor genes in the juvenile and adult of the resident population. So this is just the spread, figure 2, of all of the average mRNA expression by life stage of all 30 of those odorant receptor genes. The highest variability that we see is in olfactory, or 4.1 and 4.5 of the odorant receptor genes. So you see all of the individuals are mapped on here. The exploit river, par, and smolt way up here, way up here. The par and smolt from the Campbellton River here and here. And then even in the residents, again, we see both the juvenile and adults here, juvenile and adults here. So lots of variability. I would love to know more about what those two odorant receptor genes are doing. I believe that the information is in the supplemental information that comes along with this paper, so you can check that out at your leisure. Time is of the essence, so we're trying to get through it. And in conclusion, so we know that these genes are differentially expressed. We see a change in those expression, especially between par, smolts, and adults in those anadromous species. Populations. What we don't see is a statistical significance in upregulation or expression of those genes in the resident population, but we do see it. And he does talk about being able to put more faith in there being a change due to life history rather than genetic relatedness. So this stands to reason, right? This population of Wananichi have been isolated for almost 100 years. They're going to have some really similar genetic expression and genetic relatedness. There's a lot of interbreeding when there's only so many individuals within a population. What he doesn't talk about is the fact that in Atlantic salmon, when they're coming back, they stop feeding. Well, he does talk about this, but he does not talk about the presence of the other cells that arise as these main olfactory cells are dying or not being found in the same abundance as they were as par and smolts. So microvillus cells are used to detect nucleotides and amino acids. Ciliated receptor cells are responsible for detecting amino acids and biosalts. But this TEM image here is of crypt cells, and this is of Dania Rio. But these can be found in salmonids too, and I have found them in the salmonids I'm working on. And these crypt cells are only used for pheromone detection. And the landscape of the olfactory rosette changes as they're getting ready to spawn. So my biggest question about this whole study is, were the odorant and olfactory and glutamate and sockeye olfactory imprinting genes, were they all tied to just these two genes or these two cells? Was it just from microvillus? Was it just from ciliated receptor cells? Did we look at crypt cells at all? Did they know that there was a presence of crypt cells? And because these cells are pits, did we notice that maybe they were cells at all? There is some information about this in a paper by Hansen. I know it came out in 2003. I cannot remember, but he does go into great detail. He and she, it's Hansen and Zielinski. But they do talk about this, and that came out 7 years before this paper. So I would really need to do a deep dive to see if the suite of genes was representative of all of the chemoreceptor cells that are found in the nasal rosette. But those are my biggest questions. Let me know what you think. I can't wait to hear back from you guys about questions you might have about this. Sorry it was so clunky. Have a great day.